Hello everyone, my name is Yisu Peng. Today I'm going to talk about our work on new mixture models for decoy-free FDR estimation in mass spectrometry proteomics. Mass spectrometry is a key technique in today's proteomics and protein identification task. First, proteins are disassembled in several steps and resulting into segment ions of separate peptides, which is then being analyzed with mass spectrometry and forming mass spectra. Then it is fed into a search engine to find matching peptides from a sequence database. This search step may result in a substantial proportion of errors, which will be cascaded into later protein inference step and resulting in an even higher error rate. So controlling the error rate in the searching step is necessary, because we don't even know what are the truths, so people are using FDR to control the error rate. So we need to estimate the FDR in some manner. Nowadays, the most popular one is the targeted decoy approach proposed by JJ in 2007. It is a semi-supervised approach in which a decoy database is built to mimic the distribution of incorrect matches. The pros is that it is very straightforward and easy to use. However, it doubled the searching time. Sometimes this won't be affordable for huge databases. For example, in cross-linked peptide research, the size of the database is square and it will be a big problem to double it. Sometimes it's not easy to construct the decoy database. For example, in small molecules mass spectrometry, the structure is not in a linear chain but rather complex structures. For these reasons, we turn our eyes to another approach, the decoy-free approach adopted by Peptide Profit, which proposed by Keller in 2002. The Peptide Profit uses gamma and Gaussian mixture model to analyze scores and estimate the FDR. The pros of this method is that the model is very elegant, while the cons is that it assumes the distribution has a nicer shape of Gaussian distribution for correct matches. Here is our approach. We hypothesize that the first score is a mixture of two distributions, the scores for the correct matches and the incorrect matches. Both are modeled by a skew normal distribution. We also incorporated the set hit scores to assist the estimation. We consider the set score as a mixture of three components. The incorrect one in the top hit, another incorrect distribution that's different, and parts correct that are lower than the top incorrect ones. We derived an EM algorithm to estimate the parameters and then estimate the FDR over a threshold using the CDFs. Here are our results. This is an example dataset we take from Pride, which has about 50,000 spectra. The first figure is the gamma Gaussian model. We can see that there is some offset between the estimated distribution versus the scores histogram. On the right is the model of skew normal mixture on the top score. We can see that this model fits the data very good. On the bottom, we model both top hit and the second hit scores. Here we don't see big benefits using the set score in the model, but later we will see the difference. Here we compared the quality of the estimated FDR. We extracted spectra from NIST library, where each spectrum has an associated peptide, which can be considered as the ground truth. We compared the matched peptide against the peptide from NIST database, and then we plot the estimated FDR versus the frequency of mismatches against the NIST database. We use 4 species from the NIST and again the 68% confidence interval showing impacts. The ideal estimation is on the identical line. Above the line, meaning we are underestimating the FDR, while under the line is meaning we are overestimating it. We can see that the estimation with two samples methods is better than both the Gamma-Gaussian model and the one sample mixture. Here we run the algorithm on Hela cell experiments data. The experiments are taking different concentrations of the same components and generating spectra. Here we are showing the results of the lowest concentration experiment. The abundance of the material is very low in the sample, so it's hardly to see the distribution of the correct matches, such that the Gamma-Gaussian mixture model fails to fit the data well, but both of our models works well in this scenario. Then we did bootstrapping experiments on the Hela cell experiments. We are box plotting the estimated 1% FDR thresholds. We can see that with two samples mixture model, the estimated threshold is very stable and it keeps around at 18. To summarize, error rate control is essential in protein identification tasks, and this is also true for FDR estimation, which can be done in two major approaches, the targeted decoy approach and the decoy-free approach. Although the decoy approach is very straightforward and easy to use, there are many limitations of it. We found that in our study, the skew normal mixture model works great on the FDR estimation, and using second hit scores can improve both the performance and the stability. Thank you. That's all.